Okay, today we will have a discussion on the different type of anti-cancer drugs and their structure. So the first thing that we have already learned in our previous study or previous class is that the classification of anti-cancer drugs. So we will start from that part here. In classification of anti-cancer drugs, we learned that majority we can divide it in four to five different type of classification. However, you must remember that this is non-exhaustive, and according to different schools of thought, that these type of drugs can be classified into a few number of ways. So here we learned that first type of drugs when known as alkylating agent. So how alkylating agent works is very interesting here, and we will have a discussion. Now in case of alkylating, I am quickly telling you, alkylating agents basically denatures the DNA, how the DNA is going to have its final form in the double helical structure. That is something hampered by the use of alkylating agents. Next up we have antimetabolites. Antimetabolites are very interesting type of drugs. Here we generally starve the cells, or we can say that we generally try to poison the cells. So this is very interesting type of drugs. You will see antimetabolites, they will behave like as if they are the proper or actual nutrient, but they are not. We have different type of antibiotics which also work in different type of DNA related enzymes, just like any antibiotic works. Remember, we call them antibiotics because all the definition of antibiotics work here. They can also work in parasitical microbes, but it has been found that as they have high amount of selectivity for human cells as well, here actually we can say it is a non-selectivity. These kind of antibiotics can be used to treat cancer at the same time. We have different type of plant products. This term plant products is rather, we can say, an umbrella term. That means a lot of molecules which we have found in nature through plants, that is something that we are getting in plant products. However, you need to remember that all these plant products have different type of mechanism of action, which we will discuss in our upcoming classes. Next we have different miscellaneous type of drugs. Now miscellaneous type of drugs generally do not fit into these four different kinds of category, and that is why we are here considering them as miscellaneous. However, at different time you will see on modern classification system or in any other school of thought of classification system, these drugs are combined with other drugs of at least similar type of activity. Here for example, you can see that I have kept one drug known as cisplatin under the miscellaneous category, but this miscellaneous drug actually works as if it is an alkylating agent. However, when we will see the structure of different alkylating agents and when we will compare it with the structure of cisplatin, you will see that cisplatin has an entirely different structure when compared to alkylating agents. So in that way we can also keep it under miscellaneous section. Let's start with the alkylating agents. So I had a few annotations here. I think this annotations I will not be able to remove right now. So anyway, about all this, what I have actually written here about that, see, any cell will contain this DNA. Now DNA has two major important functions. One is cell division and another important thing is the heredity. The concept of heredity is not much important here, as much it is important for cell division. Now we know that DNA is basically a double helical strand, and these double helical strands are made up of different type of nucleotides. They are adenine, thymine, guanine and cytosine, and these type of substances are known as that is basically different type of bases, and there are corresponding nucleotides with that. These type of nucleotides will join together to create one single strand of DNA, and further these two different strands will create a double helical structure by the method of hydrogen bonding. This is something that we have here, which is known to us. Now, in case of DNA, remember that presence of enough amount of new DNA cells, new DNA molecules are extremely important for cell division. Now, in this process of cell division, what we generally find is that cell division works in a way where at first in the cell cycle you need that S phase. The S phase or the synthesis phase will work in replication of DNA. So, if you remember your biochemistry, I think you remember your replication process as well. Now, in this case, any type of DNA, when it is replicated, they first create two different type of strands, one called the leading strand, another called the lagging strand. In leading strand, the synthesis of DNA is quick, and in lagging strand it is a little slow, and there are several different type of fragments which are created, which are known as the Okazaki fragments. So, these are some basics. Now, after the formation of both the strands, they generally, by use of some kind of enzyme, DNA kinase, they will create this type of double bond. Now, alkylating agents play a very vital role here, because they intertwines the strands either with itself or with another strand. That means this type of, say, consider we have this leading strand here, and this leading strand is by some methods, what we have done is that we are using this leading strand so in a way that it has intertwined with itself. So, you know your headphones. Now, in your headphones you have one large connector which in general that goes into the 3.5 jack, but when you keep this type of headphones aside, what you will see is that they have this tendency to intertwine with itself. So, this is one example where we call it intra-strand intertwining by intra-strand entanglement. On the other hand, the two different earphones that you have in your headphone, those two strands can also get intertwined with each other in a random pattern. That is inter-strand entanglement. In this case also, suppose if there is one DNA that forces the other part to join with itself in a very random manner, which is not accepted by that cell. So, this is how an alkylating agent works. So, let us quickly learn a few things about alkylating agents. The first thing is that alkylating agents, they have the ability to form covalent linkage with most of the macromolecules which have nucleophilic centers. Now, this term of nucleophilic center is quite interesting here. So, nucleophilic center means that that portion itself will be nucleus loving, that is positive part, positive charge loving, and they will be negatively charged itself. We can say that it is an electron dense region. In this case, on later of this point, I have also mentioned here that these alkylating agents, anti-cancer alkylating agents, has a tendency to go to the N7 position of a guanine molecule. But apart from that, you must remember that there are several other molecules which also contains such nitrogen atoms which has empty or non-bonding electrons present with them, that is non-bonding pairs are present. So, many different amino acids, so we can say have that, and therefore, we can say that these kind of micromolecules have this ability to be attached with any alkylating agents. So this is one common thing about alkylating agents, thus making it very effective, but at the same time making it extremely prone to adverse effect, which can even lead to serious adverse effect of alkylating agent. The second important thing is that which we have already discussed, that this type of drugs generally damage the DNA of the cancerous cell, and they work by cross linking the DNA with one another or maybe with itself. The cross linking can be intrastrand within one strand of DNA, or it can be interstrand, that is within two different strands of DNA. And also, as I have mentioned here already, that it works usually in the N7 position of the guanine by attaching an alkyl group. So let's see some example of such a drug. The first drug here we have discussed is known as mechlorethamine. So this is how we can divide the name for your better pronunciation, at the same time for understanding that what will be the structure of that drug. In this structure, in the name itself, we understand there is one methyl group, chlorine group, and amine, that means there one ethyl group or some ethyl group with some amine group will be present. If you go back to the structure and if you study the IUPAC name as well, you will see the same kind of similarities are found here. In this structure, you can see that chloroethane group, 2-chloroethane group is getting attached or is attached with one nitrogen group, which is a tertiary nitrogen, with one methane group which is present at the same time. Let's go with the synthesis. Now this synthesis process is fairly simple, one of the simplest synthesis process in this chapter. It starts with ethyl amine. With ethyl amine, we have another cyclic group which is present, which is known as oxirane. With this reaction, we get an intermediate product where the chlorine is replaced with hydroxyl group. So obviously the name here also you will get as the corresponding hydroxyl group which is present. This is further reacted with SOCl2. You know that SOCl2 is one of the major common chlorinating agent, and thus we will be getting our final substance, which is mechlorethamine. Next up, there are some other examples as well. You can see that I have already annotated these things. The first drug here we know is known as the cyclophosphamide. Cyclophosphamide is a drug which is of the same category. You can see it also contains a cyclic group and a phosphorus group, phosphorus atom present, and the name itself is quite interesting, cyclophosphamide. Another type of drugs, actually two drugs which have very similarity with each other, and that is one is melphalan and another is known as chlorambucil. In melphalan, you can see there is one NH2 group which is present. In chlorambucil, there is one NH2 group which is absent here. Apart from that, we have another type of drug here which is unlike all these three other drugs, and that is known as thioteva. In the three drugs, or rather I would say in all the drugs, it is very interesting to see that nitrogen group, tertiary nitrogen group, which also has either cyclic or aliphatic alkyl group present, are most of the enzyme most of the important part of these drugs, and that is something that I have highlighted in all the single structure here. Next, what we have is known as antimetabolites. Now antimetabolites play a very vital role. Now we know that these type of substances, that is these DNA is made up of two important things, nucleotides, and these nucleotides are made up of two important bases, pyrimidine and purine. And in our body, purine and pyrimidine are synthesized in two different pathways. One is called the de novo pathway, another is known as the salvage pathway. We have discussed this in details in biochemistry previously. In de novo synthesis process, what we see is that these type of synthesis occurs from the very basics, and salvage means it's a recycling process. In de novo process, One major important molecule was folate, to be specific THF or tetrahydrofolate. Tetrahydrofolate is basically a folic acid derivative, and this folic acid derivative would supply some important carbon atoms in the structure of purine, and therefore making it, this tetrahydrofolate or folic acid, a very important nutrients for our body and for every single cell to have its growth continuing. On the other hand, in case of salvage pathway, which is mainly the recycling pathway, and it occurs in between the process of purine, or especially in case of purine or pyrimidine degradation. In case of purine, we all know that it degrades into uric acid, and in that process there are purine bases which are released. Now, if we have purine bases which are released, if we take those purine bases and if we treat it, that is biochemically treat it, we can get new different type of nucleotides. But what we see is that these antimetabolites type of drugs can replace both the purines or pyrimidine and the THF at the same time, thus resulting into faulty metabolism. Faulty metabolism means faulty synthesis of DNA. So, previously in case of alkylating agents, we found that those different type of strands are bound together in a spurious way. However, in this case, the strands itself are created in a wrong manner, or these are some poorly made strands. So, if we replace purine with something different, obviously there will be some changes. Now, here let us quickly check out some important facts and informations regarding antimetabolites. They generally have similar structure to those of the molecules the body use to create nucleic acid. Obviously, this is something that we have already discussed. Generally, they are low molecular weight compared to other drugs. Mostly, we can say if they are purine mimicking drugs or pyrimidine mimicking drugs, they will be low molecular weight. However, if we are talking about something which is mimicking the folic acid, such as methotrexate, they will not be very short drug or lower molecular weight drugs. Now, these drugs again mimic either the nucleic acid or important other nutrient molecules such as folic acid. The cell death is generally they will occur at the synthesis phase itself, and these drugs are mostly given for leukemia, breast cancer, pancreatic cancer, ovarian cancer, and gastrointestinal cancer. We had two examples here. One example is methotrexate. Actually, I have given two important examples here, mainly because methotrexate here will mimic the structure of THF or tetrahydrofolate, and you can see that the structure, if you compare the structure with the folic acid structure, there are quite a few similarities. And similarly, you can see that a purine like structure, that is markaptopurine, we have given the structure here. Markaptopurine will replace the use of normal type of purine, and there will be spurious type of strands which will be created by administration of this drug. So, next day what we will plan is to go with more example of different type of mechanism of action, and we will also discuss about different type of synthesis which will be important for us. So, thank you for today's class, and we will meet on the next day.